Okay, guys, so I am going to give you guys a brief lecture regarding the lab that we're going to do on bacterial transformation. And this has to do with Chapter 20 stuff that we didn't actually get a chance to lecture on due to all the snow days and stuff. So I'm going to do that right here for you right now. Chapter 20 is all about DNA technology and the idea of genomics. We're going to really focus on 20.1 in reference to our lab. And the ideas behind cloning or DNA cloning. Okay, we're in the 21st century, and DNA in the 21st century has just skyrocketed our ability to do a variety of different things in terms of genetics. So our goal of this lab that we're going to do has to do with cloning of the DNA, which simply just means making multiple copies of specific genes or DNA segments. Okay, and so what we're going to be doing is we're going to be using bacteria to actually clone or make specific copies of a certain gene, and this is going to be a gene that we get from a jellyfish, and we're going to do that all within the confines of the lab. The DNA fingerprinting we actually already saw with gel electrophoresis, so this one is really all about the gel electrophoresis lab, whereas this one is going to be, the cloning of the DNA is about bacterial transformation. Okay, so we've already done one half of the molecular genetics or molecular biology type stuff for these labs, but we're going to focus our attention on cloning DNA in the bacterial transformation lab. So we're not going to get into the details about the genomes and stuff like that, but what I want to really illustrate and to highlight here is that the more that we do, we increase our ability to manipulate the synthesis, the transcription, the translation, even the control factors of different types of DNA. And so really we're talking about DNA technology in terms of the 21st century, and that's changing lives, and it's changing science, and it's changing the way that science is perceived and taught. So in reality, when it deals with manipulating genomes, if we can look back to 1995, so this is just before, this was my junior year in high school, this was when the bacterial genome was fully sequenced. By 2003, we had the entire human genome sequenced. And so in a very short period of time, you can see that we were able to accomplish quite a lot in terms of being able to manipulate genomes, being able to read, and to essentially map out our genomes. Now, in terms of what we're doing here, we are actually focusing in on these three concepts: recombinant DNA, genetic engineering, and biotechnology. Okay, all three of these things sort of go hand in hand, so I want to talk about each of those briefly before we continue. Recombinant DNA specifically is formed when we take segments of DNA from two different sources and combine them in vitro or in a test tube or in a lab. Okay, so when we take DNA molecules from two different sources, oftentimes this is from two different species, okay, and we recombine them in a laboratory setting. Thus, the resultant DNA that we have made is called recombinant DNA. So, for example, what we're going to be doing is that we're going to be taking what we know as bacterial DNA, and we are going to join it with jellyfish DNA. Whoops. We're going to join it with jellyfish DNA. Thus, the resulting DNA molecule is actually recombinant DNA. So that's the idea here, is that we're taking two different sources of DNA, or DNA from two different sources, usually two different species, and putting them together in a laboratory setting. Okay? And so that's what recombinant DNA is. And this is also a concept, and we should really just sort of bring the idea of biotechnology up to the top here, because biotechnology is simply the ability to manipulate organisms or their components to make useful products. When we manipulate organisms or manipulate different components of organisms to make useful products. In the terms of using biotechnology and recombinant DNA, we can easily take DNA from one species, put it into a species that can rapidly divide like bacteria, and really synthesize a whole lot of certain genes or certain gene products um, in, in using this idea of biotechnology. So we've done this for selective breeding on farms. We use microorganisms to help us make wine and cheese through the fermentation process. And one other concept associated with all this is that genetic engineering has really allowed biotechnology to just skyrocket, because when we talk about genetic engineering, it is direct manipulation of genes for a practical purpose. So when we talk about biotechnology, we're really talking about the ideas of we can take and manipulate organisms and their components to generate some pretty phenomenal products. In one case, we can do this to form recombinant DNA, therefore making certain organisms make specific products for us. And the idea of genetic engineering, the ability to have direct manipulation of genes for practical purposes. Okay, so we defined biotechnology, I've defined recombinant DNA, and I've defined genetic engineering. And the ideas behind all of this stuff is that we're going to be doing all three of these things. The practice of the lab is biotechnology. We're going to be using recombinant DNA, and we're going to specifically manipulate this genome so that we can make bacteria glow with jellyfish genes. Okay, so we're doing all three of these within this lab. And the applications range from agriculture. We could take, a pesticide resistance gene and add it to our plants so that they are resistant naturally to pests, and we don't have to use herbicide, or we don't have to use pesticides. Criminal law, NCIS, CSI, all of those things are using this. We saw that within the gel electrophoresis lab. And the idea of a microarray here is that we can express all of the genes within an organism on one microscopic slide. So pretty phenomenal stuff. Really what we want to focus in on are what molecular biologists are doing. Okay, how do we find one gene within a single chromosome of DNA, and what are we going to do with that? Because one single human gene is like 1/100,000th of a chromosomal DNA. So how do we get these little tiny fragments? And really the best way to do this is to use E. coli, which we know to be Escherichia coli, which is bacteria. And we're going to use these concepts known as their plasmids. And we're going to introduce that concept here. So what we can do is we can get the E. coli, isolate these plasmids, take the plasmid, and insert that foreign DNA into the plasmid. So when we take foreign DNA and add it into the bacterial plasmids, we're essentially transforming the bacteria when we make this recombinant DNA molecule. So this lab is called bacterial transformation because it's based on the idea that we're going to take foreign DNA of a jellyfish, we're going to put it into these little things known as plasmids, and essentially when we do that we are making recombinant DNA of that plasmid. And when we give that plasmid to the bacteria, it transforms them into a recombinant organism. Okay? And we get this recombinant bacteria. Now, it's really easy to use bacteria to make clones of genes because we know that bacteria can grow and divide rapidly every 20 minutes. And so, essentially, when we say that we are using this for DNA cloning, we're really just allowing the bacteria to grow. And each time the bacteria grows and divides, one copy of that gene becomes 2, then 2 become 4, and then 8, and then 16, and 32, and so on. So these clones of bacteria, as they clone each other, they're actually cloning the foreign genes at the same time. All right, so what are these plasmids? Well, E. coli has a typical normal circular chromosome inside of it that has its basic genes that it needs. But bacteria also have these really awesome things known as plasmids. Okay, and plasmids are small circular DNA molecules. They're smaller than the bacterial chromosome, and they can replicate separately from the bacterial chromosome. And these are extraordinarily significant because they contain a small number of genes. Okay, so these plasmids contain a small number of genes, And they can be used by bacteria only when the environment dictates that they need to. In other words, these plasmid genes tend to become useful when the bacterium is in an environment that requires it to transcribe and to translate the plasmid genes. So essentially, the bacterial chromosome is used for basic metabolic functions, whereas these plasmid DNA, these small circular plasmids of DNA, can be multiplied and grown inside the bacteria on their own. And these plasmids have specific genes that are only going to be transcribed and translated when the environment dictates or when the environment selects that they should be. Okay? So cloning is really important because we're going to use these plasmids. If we take a plasmid, insert a foreign gene into it and make it a recombinant DNA plasmid, and then insert that into the bacteria will then clone themselves, but they will also be cloning that gene that we had put into the plasmid. Okay? So we copy a particular gene for a particular gene product. We're giving organisms new metabolic capability. Okay, and so the question is, metabolic purposes or medical purposes, any others? Our goal in this lab is going to be to take regular E. coli, transform them by giving them jellyfish DNA. We're going to put jellyfish DNA in the plasmid. Then we're going to insert the plasmid into the bacteria to form recombinant bacteria. And then we're going to just let them grow and clone those genes. And then, of course, what we're going to do is select from those clones to see which ones successfully took up that plasmid. Oh, did I go too far? Okay, so this is the basic technique without all the details, but we're going to briefly talk about the details in a second. So you can clearly see that if we take, and so our ideas here are, and I'm doing this specifically because I want you guys to pay attention, magic pen. Okay, so here we have the bacterium, okay, and you can see it has a bacterial chromosome and it has a plasmid. So what we can do is we can remove the plasmid from the bacterium and we can take a gene from something else. So here is our other cell containing a gene of interest. We're going to remove that gene and we're going to insert it into the plasmid that we got from the bacteria. Okay? So this plasmid is now actually a recombinant plasmid. And then what we do is we take that recombinant DNA plasmid and we're going to put it back into the bacteria. So this bacterium is now essentially considered a recombinant bacterium. We're then going to allow the bacteria to grow and divide and to essentially clone itself, but at the same time clone that gene. And of course, the gene of interest for us is going to be, we could pretend that this is a jellyfish. And the gene of interest is going to be, I think we're going to call it either P green or P glow. It doesn't really matter to me how we talk about it. But the point is it's the gene for bioluminescence. And we're going to put that into bacteria so that eventually the bacteria would be able to glow. Now, of course, in this diagram here, it's showing that we could put anything in here. We could put human growth hormone. We could put human growth hormone. We could put a protein that dissolves blood clots that for heart attack therapy. So a lot of the medications that we have for individuals who have had heart attacks, we can generate those proteins and make the pills for them. We can insert a gene that allows them to clean up toxic waste, or we can put a gene that allows them to resist pesticides. So you can clearly see that this is a significant process in terms of what we're going to do. So one last thing I want to talk about before I actually break into the idea of what the lecture or what the lab is all about is that in order for us to make recombinant DNA, we have to be able to cut the plasmid DNA and our target DNA using the same enzymes. And remember, we talked about these enzymes known as restriction enzymes, because what restriction enzymes do is that they are targeting specific sequences of the genome. These were discovered in the 1960s when individuals were studying bacteria. These enzymes are used by bacteria to cut up foreign DNA that a virus, or what we call a bacteriophage, would inject into them. So these enzymes' source are from the bacteria, which is pretty awesome. We talked about that. We had EcoR1, we had HIND3, all those different ideas. Okay? But what the restriction enzyme does is that it's an enzyme that's going to cut DNA. And there are hundreds of different enzymes that are restriction enzymes, and each one can cut a specific site of the DNA. Okay? So these restriction sites are short sequences of DNA recognized by the restriction enzymes. Okay? So these are short DNA sequences that are recognized by the restriction enzymes. Okay? And these restriction sites typically occur multiple times within a genome. Okay? And so you can see the examples up here. You can see that this is a restriction site up here, and that the enzyme is going to come in. This restriction enzyme will specifically cut, and in this case it's cutting, I did that wrong. It's cutting at a jagged angle. Okay? Not all enzymes do this, but typically the ones that we're going to be using, or the ones that were used to generate recombinant DNA, are going to cut at this angle. Okay? And it ends up making multiple different DNA pieces, and each fragment of DNA that was cut by the restriction enzyme, when it cut at specific restriction sites, we end up calling them restriction fragments. So a restriction enzyme is going to target a specific restriction site. These restriction sites occur multiple times in any given genome, so that when we apply the restriction enzyme, we end up with multiple restriction fragments of that genome. Okay? So what we can do is we can take these restriction enzymes and cut them in such a way that we end up with these things known as sticky ends. The sticky end is simply bases, okay, single-stranded pieces on the double-stranded DNA that result from the restriction cutting. Okay? And they are called sticky ends simply because they want to make hydrogen bonds with complementary other sticky ends. Okay? So this little single-stranded end is called a sticky end because it wants to hydrogen bond with other sticky ends. So what we actually get to do is we could take, so for example, we're going to take the bacterial plasmid DNA, okay, the bacterial plasmid is going to be cut with the same restriction enzyme as we cut the jellyfish genome, specifically for the P green or P glow gene. So we cut the bacterial plasmid with a restriction enzyme. Let's say it's EcoR1. We're going to cut the jellyfish genome with the same EcoR1 so that we will have matching sticky ends from the two different sources. So when we have the DNA fragment from two different molecules cut by the same enzyme, these sticky ends naturally will come together, okay? And we can actually insert what we'll call this one the P green gene. We can insert that into the plasmid. And then, of course, the glue that reanneals or puts together the sugar phosphate backbone is known as DNA ligase. And we learned about DNA ligase where it put together the Okazaki fragments when we were talking about DNA synthesis and transcription, or DNA synthesis a few chapters back. So that's the idea. Those are the ideas behind the lab. Now I'm just going to sort of bring you up to speed as to what we're going to be doing specifically in the lab while I'm continuing this talk. So that's the introduction aspect. So specifically in our lab, what we're going to be doing is we're going to take our E. coli, okay? So here's our E. coli. Whoa, sorry. So we'll get rid of that. So here's our E. coli, and here's its bacterial chromosome, okay? And what we're going to do is we're going to chemically shock them using calcium chloride to poke holes in their cell walls and plasma membranes. So we're going to take non-transformed E. coli, chemically poke holes in them so that that cell could receive foreign DNA. We're then going to add this little plasmid DNA that I'm going to have in a vial. I'm going to use the micropipette. So essentially we're going to take this and we're going to add it because hopefully it will be taken up by the bacterium through those holes. So this is the transformation part. And what's actually on this gene, and this is just an abbreviation of what it would be, but here's the whole plasmid, and on this plasmid are going to be two different genes. We've got the pea green gene from the jellyfish, because if that gene here gets transcribed and translated, the bacteria should be able to glow. But we're also going to put the gene for ampicillin resistance. Remember that the plasmid genes are only going to be transcribed and translated should their environment dictate that they need to transcribe them. So by adding this to our plasmid, this gives us an ability to select for and against different bacteria that have actually taken up the plasmid. So what you need to understand is that each plasmid that we're adding is going to have the glowing gene, pea green, and a gene that allows the bacteria to be resistant to ampicillin, which is an antibiotic. So we add the plasmid, hopefully some of them take it up, and then we're going to put the bacteria on ice for 15 minutes. Because this is going to kind of slow them down, kind of let them recuperate from this process. Immediately after, we're going to heat shock them. So we're going to cool them down, then we're going to heat them up. And heating them up pretty much means, hey, you've got all these holes in you, it's time to repair them. Okay? So by putting them in the ice and then shocking them with heat, it's going to trigger these bacteria to repair any holes that they have. So some of the bacteria may have taken up this plasmid. Understand, guys, that we're on the orders of 10 to the 10 to 10 to the 18 bacteria that could take up the plasmid. In reality, we may get 1 to 10 individual bacterium that become transformed. Okay, and it may be even lower than that. The point being, very, 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 very few bacteria are successfully transformed. So we're going to have to select for them. So we heat shock them in water bath for exactly 90 seconds, and then they should have repaired their membranes, and then we're going to give them food. LB is known as Luria broth. A lot of times it's like what you would get in those little, like the stuff that come in ramen noodles, like the little brown packets, like the little, the chicken bouillon, those little bouillon cubes. Same idea, Luria broth, it's just food for the bacteria. And when you feed your bacteria, it makes for happy E. coli. Obviously, the little eyes are just added for fun. It's not actually any new plasmids, okay? So, of those billions and trillions of bacteria that we have in the culture, maybe a handful of them actually have taken up the plasmid. So then we feed them, then we put them in the incubator, okay, and we're going to put them on agar. Okay, and one thing to understand here is that the agar is like this gelatinous substance that contains the Luria broth. So this agar is what's going to be in the Petri dish. So if you're looking at the Petri dish, okay, that little layer, okay, at the bottom, that's like this brownish, weirdish like layer, that is actually the agar. Okay, and so in the agar is this Luria broth. Okay, so that's the idea behind the agar. So we're going to feed them, but then we're also going to put them on this gelatinous substance that has the Luria broth in it. And hopefully with heat and food, we're going to have a whole bunch of happy E. coli cells. Now these groups of E. coli cells are going to form what we call a colony. So each of those would represent this guy right here, like that, like what I just showed you. So everything over here would actually constitute one of these little colonies, okay? When we are plating the bacteria, we could get colonies, which are little individual groups of bacteria. We tend to get colonies when we are selecting for only the bacteria that are transformed. In other words, we're going to add billions of bacteria to our culture plates, but only a few are actually going to be transformed. We tend to get colony growth when we are selecting for the transformed and against those that are not transformed. We tend to get a bacterial lawn when there's unrestricted growth, okay? i.e. no selection. And so for our lab, we have 4 culture plates, okay? One of the culture plates, there will be, in the agar, we are going to have food. Whoops. So in the agar, we have food. But in the culture that we're going to add to it will be bacteria that do not have the plasmid. So the plate has food. We're going to add bacteria that have not been given the plasmid. We should expect to see a lawn growth because we're not selecting for or against anything. We're also going to plate on the Luria broth agar. We're going to add the colonies that we've made that we've given the plasmid to. But because we're not really selecting for or against anything, we should expect to see lawn growth as well. This is just to test whether or not the transformed bacteria grow as well as the untransformed bacteria. Okay? In the other 2 plates, we are going to have along with the Luria broth in the agar, we're going to add ampicillin, which is an antibiotic. So that if we add any bacteria onto the plate, they're going to be exposed to the ampicillin antibiotic. When we add the culture that was not given the plasmid, that means that they do not have the ampicillin resistance gene, and they do not have the P green gene. If they don't have the plasmid with the ampicillin resistance, they can't grow. So there would be no growth on this plate. But if we take the LB broth and the ampicillin in the agar, and we add the colonies that we added plasmid to, only those that have the plasmid will be selected to transcribe the ampicillin resistance gene, to be resistant to the ampicillin, to grow. But because it was also transcribing the ampicillin resistance gene, it's also going to transcribe the P green. So on this plate, I usually say growth and glowth, which is a stupid word, but it rhymes. No grow, no glow. Okay, so review this. This is going to be extraordinarily helpful for the lab, and I can't wait to do this lab with y'all because it's a lot of fun when it works. So review this a couple times if you need to, and hopefully it is enough to help you understand the lab.